Okay, hello everyone, and thank you for joining us for today for General Session 2 of the Simons Searchlight Virtual Conference. Before we begin, I just want to let everyone know that you're able to submit questions at any time during the presentation using the Q&A button at the bottom of your screen. As you submit questions, please try to refer to the presenter or presentation topic that the question is for. And please be aware that we may not have time today for all questions, but we will do our best and we'll follow up at the end of the meeting to address any questions we couldn't get to. And with that, we'll begin the presentations.
Hello, and welcome to Simons Searchlight and Research 101. My name is John Spiro. I'm a neuroscientist and the Deputy Scientific Director at the Simons Foundation. Today, we're really excited to present a series of short talks on Simons Searchlight and also some background on the research activity being carried out around the world. Our goal at the Simons Foundation is to support research that will help improve the lives of families like yours. And our goal with these talks is to give families a sense of how this research happens, and importantly, to give you a chance to ask questions. Here's a list of today's talks. First, there'll be a talk, what is Simons Searchlight, how it works, and why its participation is so important. This will be given by Dr. Misha Kawanda and Dr. Leanne Green-Snyder. Then I'm going to come back and talk about what is the Simons Foundation and the Simons Foundation Autism Research Initiative. I'll talk a little bit about animal models and why they're so important. Then my colleague, Dr. Julia Sommer, will talk about what are induced pluripotent stem cells, or IPSCs, and how do they fit into the bigger research picture. Then my colleague, Dr. Wendy Chung, will talk about what types of targeted treatments might be possible in the future. And finally, my colleague, Dr. Paul Wong, will talk about how clinical trials work and what are some of the challenges. Importantly, then we'll come back with a panel and give you a chance to ask us questions.
Hi, my name is Misha and I'm the Outreach Manager for Simons Searchlight. Today I'll be presenting along with my colleague Dr. Leanne Green-Snyder to give you an overview of the Simons Searchlight Research Program. Simons Searchlight is a research registry for individuals with genetic conditions associated with autism and other neurodevelopmental disorders. Many of them are very rare. In this presentation, Leanna and I will walk you through what it means to participate in Simons Searchlight, what we are collecting for research, and how researchers access participant data. By the end of the presentation, we hope you walk away with a clear understanding of our process, the security regulations we have to protect our participants, and the importance of collecting high-quality research data as we aim to accelerate research into these genetic conditions. Simons Searchlight's mission is to help move genetic research forward by collecting detailed information from participants, helping link participants with others who have the same condition, working with researchers to identify what information should be collected, and freely sharing this information with the scientific community. We believe it is critical to keep the data open to the research community, both basic research scientists, those in the lab who don't normally work with patients, and clinical researchers. This December marks our 10-year anniversary to this program, and our efforts are committed long-term in this area. Simons Searchlight is funded under the Simons Foundation Autism Research Initiative, or SAFARI. Dr. John Spiro will be giving more detail on SAFARI in his talk. So we see our goals of Simons Searchlight under these 5 categories. First is to collect medical and behavioral information along with blood or saliva samples from participants. Our second goal is to provide information back to the Simons Searchlight community about what we're finding. And some examples of this are the individual result reports uploaded to your dashboards, the presentations we give at family meetings specific to your genetic condition. We know that providing information back to you helps you navigate your day-to-day, whether that be working with your medical provider, communicating with your school, or just feeling more informed about what you can expect. Our next goal is to share everything provided by participants, so our samples and the information we collect, to qualified researchers in a de-identified way. We also aim to connect participants to others around the world, either informally or through dedicated Facebook gene pages. Finally, we have this overarching goal for all these efforts, and it is to promote better understanding of these genetic changes. More information and more understanding will lead to faster progress. The genetic changes that we study in Simons Searchlight include over 200 conditions. This list is reevaluated quarterly and has grown substantially over the last several years. We do not have all the participants enrolled in all these genetic conditions, but we expect to continue to grow as we grow over time. Participation in Simons Searchlight starts with just a few steps. As a participant, you can sign up online wherever you live around the world, currently in English or in Spanish, but there's more languages to come. After uploading your genetics lab report, which is step 2, you schedule a phone call with our genetic counselor, which is step 3. Genetic counselors are medical professionals who guide you through a set of questions asking about you or your child's medical history. The next step, step 4, is we assign online surveys that provide additional important information for researchers. We build new surveys regularly, and we follow up on your medical history yearly. In the final step here, we note that there are also multiple ways that we help connect other researchers with participants or their data. Dr. Green-Snyder will now talk more about our surveys and how we work with researchers.
Okay, great. Thank you, Misha. So I'm going to talk about the kinds of information that we collect in Simons Searchlight and why we collect it and what do we do with your information. So on our side, we call the medical and behavioral and developmental information we collect, we call that data, of course. Phenotypic data is everything that we observe about an individual, their behavior, their development, and information we know from the medical history. So we try to collect everything that researchers need and want to know to fully understand your family's genetic change. We follow participants over time using what's called a longitudinal follow-up so that we can track the natural course of the condition. We use the higher standards of data collection, emphasizing consistency in measurement and established scientifically validated psychological measures. And we do this to be able to look at severity and type of the phenotype associated with your genetic change, and to ensure we're able to compare across individuals, compare across groups, and to compare over time. And finally, so researchers are able to combine our data potentially with other patient groups, even throughout the world. We also incorporate your input and researchers' input for what's important to measure. So for example, very specific medical concerns. We have two kinds of data listed here, two kinds of measures. So in terms of medical records, we review your clinical lab report. And in terms of telephone interviews with a provider, with a professional, we do the medical history, we do a developmental and a diagnostic history, and we do an adaptive functioning interview that essentially gives us an estimate of development, and that's called the Vineland Adaptive Behavior Scales. We also have online measures, and the online measures are measures that you fill out. So you report to us background characteristics, demographics. We have a very special survey that's a detailed seizure history survey. The validated measures, which are another type of measure, they're licensed and copyrighted and purchased from publishers. And these were developed over years by researchers and shown to validly measure behavior and different kinds of behaviors and communication. Those are here, the examples are the Child Behavior Checklist, Social Responsiveness Scale, and Social Communication Questionnaire. Examples of some of the measures that we have authored with our partner researchers or our clinical researchers, physicians on our staff Examples are skin and dental surveying, the learning to read survey. And we have more surveys to come that we are creating. We make sure to update these measures based on the latest research and the best practices in the field. So for example, we're updating to the newest Vineland Adaptive Behavior Scale soon this year. And we also adapted a survey from the Rare Epilepsy Network survey so that we could use the cutting edge in measurement for epilepsy. We conduct very detailed medical histories. Those are conducted by certified genetic counselors, as I mentioned earlier. This ensures the highest quality data. We return your data to you, and this is how we do that. We make sure to share the data back in customized reports that are about your child or dependent for whom you filled out information. And so this is an example here, a sample from the Vineland Adaptive Behavior Scales. And we send back not only how the cohort is doing, but where your child lands in comparison to others in the cohort. We just changed website providers in the last year, and so we're in the process of reactivating these reports for you right now as we speak. And so a very large batch of these will be going out soon. We also strive to make direct contributions to the body of knowledge out in the world about your family's genetic change using your data. So these include genetic reference guides. There are a couple examples here. Also scientific journals, so actual studies making discoveries about the nature of the genetic change, and presenting to international scientific conferences. We also collect biospecimens. We collect two types, blood and saliva. This is optional for you, but if you choose to participate, we store them in a biobank and we make them available to researchers. Researchers use these biospecimens to develop cell lines, to study how a gene operates inside of cells, and to develop potential treatments. This is extremely important because researchers have a very difficult time finding patients and samples since these are rare diseases. So what we're able to do is to provide power in numbers and to provide this resource centrally from one place to researchers all around the world. This is a very unique resource because we also connect every biological sample to detailed medical and developmental information. So if you donate biospecimen, the researcher will have a picture of the whole patient. And Dr. Julia Sommer will talk later about developing cell lines from these biospecimens. They're incredibly important because deeper genetic analyses are needed to understand individual differences. Here are some of our resources that we offer to researchers around the world. Your phenotypic data, that is what we covered with medical and developmental information, genetic data, imaging data for some cohorts, the biospecimen, and now a research matching service. ResearchMatch is a resource in which researchers can apply to connect with you so that you can participate in further research into your genetic change. And we will discuss that a little bit more later. Now, for our resources to host our research registry and for our partner researchers to use the Searchlight data, all of us are required to have approval by an IRB. And some of you have asked us, what is an IRB? So we're going to review that very briefly. The IRB is an institutional review board. It's an independent ethics committee, and it's required by federal regulations by the US Department of Health and Human Services to review all research involving patients, to ensure the protection of the rights and welfare of participants, and to ensure the proper use of data. All research with people is required to have this kind of review, including biotech companies that may be developing treatments. Also, a researcher's university is requiring that the researcher have their proposal reviewed to determine how much more review is required to make sure the research is done properly. These IRB regulations are there to ensure that you know where your data is going and what is going to be done with it, and that it's being used properly and ethically. So if someone were to host a collection of patient data to be used for research, or even if just a researcher wants to use patient data in a registry, if they are not reviewed by an IRB, they are violating federal regulations. So we at the foundation make sure that Simons Searchlight has all approvals for our registry and also for the researchers that want to use our data. In fact, we're approved by 2 separate IRBs. Here's an overview. This is what researchers see in a description of how they will access your data. So this is simply how they come to our website. It's base.safari.org to request data, and it's very simple. So they come here, they create an account, they make an application with their IRB approved project, they register their university, they sign a researcher distribution agreement with us that promises they're not going to sell your data or send it around to any unauthorized parties, and that they will share the data back with us so that we can share it with other scientists in the future and pay it forward. And this is what it looks like to the researcher when they come to our website. This is what Safari Base looks like inside. And so they literally, once approved, can check off the data sets that they would like to request. So down below, you see there's a Simons Searchlight longitudinal data set that they can select and download. And also, this is the place they would go for ResearchMatch applications. And ResearchMatch is a new free recruitment service for researchers to match with you, the families, to do new studies. These could be studies focused on understanding a particular medical concern more deeply, or it could be for future treatment studies. And the researchers who use ResearchMatch also are asked to share their data back with us. So again, the cycle goes on and we can share it on with other researchers.
So for the last slide, we want to finish on what we identify as the power of our large genetic registry. One of the strengths of a unified research registry like Simons Searchlight is that the effort made for one genetic change can usually help others. What I mean by this is that by finding the best surveys that we can use to measure a condition, such as seizures, we can offer this to all the applicable conditions. And this is done as example with this new seizure survey that was adapted from the Rare Epilepsy Network that Leanne was talking about. Many of our neurodevelopmental conditions have seizures as a feature associated with their genetic condition. Another example of the power behind this research registry is that a researcher can come in and study different genes as a comparison to others. As shown in the example on my slide, gene 2, gene 3 have some similar features and some different features. Why this is can be used by researchers to learn more about both conditions, or researchers can study other genetic conditions on our list as a group, which they might be acting in the same genetic pathway. So thank you for listening today.
So I'm back. Here we are in the agenda. I'm going to talk about what is the Simons Foundation and the Simons Foundation Autism Research Initiative, and why animal models are so important. I'll give you some background on the types of research that we support and some information on why animal models are an important step in the path to thinking about targeted treatments. The Simons Foundation is a New York-based private foundation. It was founded by Jim and Marilyn Simons. Jim is a mathematician turned investor turned philanthropist. And the broad goal is to advance the frontiers of research in mathematics and the basic sciences. The foundation has a very big endowment and a large annual budget in the range of $500 million. The point I want to make by mentioning that is that the foundation will be around for a really long time and is committed to long-term research projects. And that long-term commitment is really critical to work in this field. Within Simons Foundation is a group entirely focused on autism and related neurodevelopmental disorders. Everything that I will mention today is supported by this division, which is called SAFARI, which stands for the Simons Foundation Autism Research Initiative. Safari has been around for about 17 years. The annual budget this year is about $90 million. And one of the biggest roles of Safari, as you heard in the previous talk, is to fund the creation and curation of large cohorts of research subjects. This includes Simons Searchlight. I want to emphasize here that all of these cohorts, all of these research projects are open community resources. Our hope is to encourage the best researchers from around the world to work on the very challenging problems of neurodevelopmental disorders by making great high quality data readily available to them. Safari also makes grants directly to researchers. They come in a few different kinds. You don't really need to know too many details here, just to mention that they can be pretty large, and we support a lot of investigators. Since 2003, Safari has supported over 580 independent investigators. We fund work all around the world, and we emphasize quality and rigor. All the work that we fund is evaluated by a very rigorous process that involves multiple levels of peer review. One of our goals, and the things we're really proud of, is that we've brought many new researchers into the field. We've lowered the bar of entry by putting out grants, tools, and resources. Now I want to switch gears a bit and talk in a bit more detail about the type of research that we support. In particular, once we know the gene that causes a condition, where do we go from there? The first thing we can do is learn as much as possible about the human biology. Parts of this were covered in the previous talk. And this starts really with a detailed medical history. We try to gather as much data as possible to understand how people who carry a given genetic variant develop, what kind of challenges they have, and we do this at multiple points to try to understand the development. A big deal here with the so-called genetics first approach that Simons Searchlight takes is that instead of studying a group of people that is defined by a given set of behaviors like autism or developmental delay, we can study people who in theory all have the same causative genetic event, and that's illustrated here. So instead of studying a heterogeneous group of people, we're trying to study people that all have the same causal event. Our hope is that this will lead to much clearer conclusions compared to studying people with that share, for example, a behavioral diagnosis, which might be the result of a large number of different types of causes. The next thing we can do is to use some of the amazing instruments that have been developed to look at the brain using non-invasive technologies. These include MRI, EEG, and MEG. And researchers using these technologies ask questions about the overall structure and function of the brain and brain activity. So here's an example, you've probably seen pictures like this, of an MRI, that magnetic resonance imaging. These are pictures of the brain taken totally non-invasively. There's no, doesn't hurt at all. And you can see the brain in rather exquisite detail. An encephalogram, or an EEG, measures brain activity with electrodes placed on the scalp. And you can see some examples here of how brain activity changes during wakefulness and different stages of sleep. An MEG gives activity very much like an EEG, but allows some different levels of resolution. A third approach is to generate and study animal and cellular models that can sometimes be engineered to study the same specific genetic variants that are found in people. On this slide, I'm showing a picture of some of the most common organisms used in labs around the world. These include the worm, the fly, fish, mice, rats, and a type of cell called an induced pluripotent stem cell. I'm not going to say any more about these because my colleague, Dr. Julia Sommer, is going to talk about these in detail next. Now, you might think I'm crazy. I just showed examples of imaging technologies on the last slide that allow researchers to study people. Why on earth would someone turn to an animal model, and especially something like a worm or a fly when they look nothing like people? Well, it turns out that what we really want to understand is how changes in any particular gene can lead to changes in behavior. But genes, shown here as a strand of DNA that makes up a chromosome, which itself sits inside the nucleus of a cell, genes don't code for behaviors. Genes code mostly for proteins. They make up our cells and perform all sorts of functions within our bodies, which in the brain in turn make up circuits and systems that control behavior through very complex processes that we are just starting to understand. So to really understand how changes in any particular gene matter, we often want to understand how those changes influence the cells and circuits. And to do that, we need to use systems that we can study in great detail. And those are usually animal models. The imaging technologies that I showed previously can measure brain activity at sort of a 30,000-foot level, so to speak. But we often really need to get into the details and manipulate experimental conditions to really learn how cells and circuits are changed with changes in genes. And importantly, this type of understanding is fundamental to thinking about rational and targeted therapeutic interventions. By the way, shown here is a close-up of a part of the brain called the hippocampus, which is involved in memory. And if you look closely, you can see the incredible amount of detail in the individual nerve cells, these processes here, and you can see how they're connected in very complex ways. Sadly, the real brain doesn't have these beautiful colors. That's added after the experiment. So what are some of the key research questions that we want to answer in order to think about rationally about targeted treatments? And which animal model are good for which type of questions? Well, some of the most basics of these questions can be answered in the worm or the fly. It turns out that the worm is about 40% genetically similar to humans. So many of the genes that are in our own cells are also in worms, which, not surprisingly, are far more experimentally tractable than humans. What this means is that we can quickly and relatively cheaply design experiments to answer questions about what a given gene does, and for example, what other genes and proteins it interacts with. One of the types of experiments that we can readily do in fish, which are about 70% genetically similar to humans, is to remove any given gene using some molecular tricks, and to ask how that changes cells or circuits of neurons. Does it matter when and where the gene is deleted during development? A great advantage of fish, this particular fish is called a zebrafish, that's commonly used in labs, is that it has very clear skin, especially when it's young. So researchers can image individual brain cells directly through the skin very readily. Similar types of questions can be asked in mice and rats, which are even more genetically similar to humans, usually around about 80%, though the experiments here become more costly. A key point that I want to make is that researchers are not making a model, per se, of human disease. Many human diseases are really too complex and human-specific to ever really model in another species. But we're using these animals to try to understand molecular and cellular mechanisms that result from changing a gene that is indeed shared between the experimental animal and the human. I hope that distinction is clear. As a side note, I'd like to mention that one of Safari's goals is to make these experimental animals. I talked about, I made it sound like it's easy to engineer these with genetic changes that resemble changes seen in humans, but it's not really easy. It takes time and money. And so one of our goals is to make these experimental tools available to the research community so that we can get the best researchers around the world to think about these problems. So we've established various types of repositories where we and the researchers that we support deposit these animals so that any researchers around the world can use them. On this last slide, I want to give you one example of a type of experiment done in one of these experimental systems, in this case, a mouse, that allowed us to learn something that could not have been learned from humans, but might have profound implications for thinking about targeted therapeutics in people. The experiment was published in 2007. This is actually a picture of the paper itself that appeared in the scientific journal Science. This has become a really famous paper, so I just thought I'd show you what it looks like originally. And this had a really huge impact on the field of neurodevelopmental disorders. Investigators made a model of Rett syndrome in a mouse. Rett is a severe neurodevelopmental disorder characterized by motor and breathing issues, anxiety, and cognitive problems that include autism spectrum disorder behaviors. And it's caused, we know, by mutations in a gene called MECP2. In this case, the mouse model mimicked many features of the disorder. So the mice also had motor and breathing issues and cognitive deficits, much like the people who have Rett syndrome. In this very clever experiment, researchers generated a mouse model where the gene had an engineered molecular switch, which allowed investigators to turn the MECP2 gene on or off, depending on whether they fed the mouse a very specific compound. The details of exactly how this worked experimentally are not really critical here. So for now, just understand that they could make the gene active or inactive when they wanted to. This just shows you that when the gene is off from birth, the animals develop symptoms at around 4 to 7 weeks after birth, and then quite severe symptoms after that. And what I'm showing you on this next slide, and this was the really really shocking part of the experiment, was that even after the onset of symptoms, when the gene was off, if you then turn the gene back on, the animals, this axis shows how severe the symptoms are. Up here means not very severe. Down here is very severe. So the animals are going along here with the gene off. If the gene stays off, they start to decline and their symptoms become much more serious. But if you turn the gene back on, again, after the point when many of the animals already have severe symptoms, you can actually maintain a relatively healthy animal. So this was a really surprising result. It showed that a supposedly neurodevelopmental gene had ongoing effects after birth, and critically, that a late rescue was possible. By turning the gene back on late, you could rescue some of these symptoms. This is really quite an encouraging result. So a key question is, how many more neurodevelopmental genes are like this? Almost certainly not all, but maybe more than we would have thought. Of course, this is work in a mouse. Could such a gene replacement strategy work in humans? My colleague, Dr. Wendy Chung, will discuss this fascinating topic in a subsequent talk. Okay, I'm going to stop here. I look forward to your questions at the panel at the end of this talk. The next speaker will be Dr. Julia Sommer, who will discuss an amazing emerging technology where we can create brain cells directly from blood.
Hi, my name is Julia Sommer, and I'm a scientist on the Safari team. Before joining Safari, I did research in molecular biology and neuroscience. Today, I want to talk to you about induced pluripotent stem cells, also known as iPS cells, and why they're special. And I also want to tell you a little bit more about how researchers use iPS cells, and then talk about the Simons Searchlight iPS cell repository. So as others have mentioned already, individuals who register for Simons Searchlight have the option to donate blood for research. And this can happen remotely through Quest Labs in the US, but also at in-person family meetings where international participants can donate. The donated blood is sent by mail to our biobank at Rutgers University in New Jersey. And the blood is used to make 2 types of research resources. One is a cell line that we won't talk about today, and the other is induced pluripotent stem cells, or iPS cells. And in my next slide, I'm going to talk a bit more about how iPS cells are made and why they're valuable. So the iPS cells we are making from Simons Searchlight participant blood donations are made from blood. But iPS cells can also be made from skin cells, but we only collect blood in Simons Searchlight. Within your blood, there are many different types of cells, and researchers isolate those that they want to use to make iPS cells and then treat them in a special way to force them to become so-called iPS cells. This first step here shown takes about 6 to 9 months. And I wanted to point out that making the iPS cells is actually not an experiment, despite how long it takes. Making iPS cells is really generating a resource that researchers can use to do specific experiments. By making this resource, we hope to save researchers time and money so they can focus on real experiments. But the question is, why are iPS cells so valuable? iPS cells are very special because researchers can force them to become the specific or the cells that make up the heart. So those cell types are all different, and importantly, they are not easily accessible from a living human. Blood cells that were not converted to iPS cells cannot turn into these different cell types on their own. The only other cell type that can develop into such specialized cell types are embryonic stem cells, which are not available from patients. So that's why iPS cells are so valuable, because they allow us to make and study usually inaccessible cells from a patient. So researchers can then use those nerve cells, for example, to understand how and why the cells of individuals with neurodevelopmental conditions function differently from those of other people. They can also be used to find and test drug therapies. Those iPS cells to study neurodevelopmental conditions. As briefly explained on the previous slide, researchers can force iPS cells to become nerve cells, which means it allows researchers to study the behavior of brain cells of a living human being. That's pretty amazing, actually. Here is an image of some real nerve cells grown in a plastic cell culture dish, and researchers stained those with a special dye so they're more easily visible under the microscope. So what researchers do is they make nerve cells from patients' iPS cells and nerve cells from control iPS cells. Those control iPS cells can be those made from a family member's blood, or the control iPS cells can be made from the patient themselves, but researchers used a genetic trick to repair the genetic error that is found in the patient. Now research can compare all these resulting nerve cells here to each other and learn what's different between those from patients and controls. Things they may look at, for example, are the shape of the nerve cells. Nerve cells have these very special shapes with long branches, and it's possible that patients' mutations affect the ability of nerve cells to take on that specific shape, which can affect their function. Researchers may also test with each other by looking at the special junctions called synapses that nerve cells form among each other. Again, if patients' nerve cells don't form those connections as well as controlled nerve cells, it may cause them not to function properly. Lastly, they may also look at the electrical activity of the nerve cells. Nerve cells communicate by sending small electrical signals down their branches, and this could also be disrupted by a patient's mutation. If researchers do find that the nerve cells from patients are different, they can use the nerve cells to test different drugs in the hope to find drugs that remove the difference and make patient nerve cells look and act the same as control nerve cells. The hope is that drugs identified in that way would also repair the nerve cells in a patient's brain and not only those grown on a plastic dish. However, as always, things are not quite as easy as they sound. And after all, the nerve cells grown on plastic dishes, and they don't grow exactly the same way they would in a human brain. So there is actually a good chance that drugs that work under this condition won't work in a living human. And that's one of the reasons why researchers use many different ways to look at nerve cells made from patients' iPSCs. Another way to look at them is by growing the nerve cells into something called brain organoids. In a brain organoid, nerve cells, instead of growing next to each other on a flat plastic dish, grow in a three-dimensional arrangement. they grow on top of each other, next to each other, and so on, much more in the way they're organized in the brain. Although it's important to remember brain organoids are a long way removed from resembling a real human brain. Brain organoids can be grown for multiple hundred days. They kind of look like tiny tissue balls, and here is an image of some real brain organoids. And again, researchers can look at several properties of the cells that grow inside those tissue balls to understand what's different between patients and controls. And they can, again, for example, look at the shape of the nerve cells in those balls. They can look at their connections and their activity. They can look at the number of the cells in the balls and even their size themselves. And because the nerve cells in the brain organoids develop and assemble more in the way they would in a developing human brain, the hope is that drugs that are discovered in this way are more likely to work in a living human. In addition, the nerve cells in the brain organoids, as compared to those that are grown on the flat plastic dishes, more closely copy the individual steps that happen during development in the real human brain. And that raises the hope that they will give a more accurate picture of what and when things may go wrong in neurodevelopmental disorders. I wanted to emphasize one more time that brain organoids, even those grown for many hundred days, lack many attributes of a complete brain. So they're not mini brains, despite what they're frequently called in the media. So the power behind a project like Simons Searchlight is that researchers can get access to iPS cells and clinical information from the same individual, as well as a large group of individuals that share causes for their disorder. This helps researchers to get a more complete understanding of each single individual, but also allows them to learn what all those individuals have in common or what sets them apart. And what's more, researchers have access to iPS cells and clinical data across multiple genetic conditions linked to autism and neurodevelopmental disorders. And we hope that the easy access to iPS cells and data for multiple genetic conditions will make it easier for researchers to connect and compare across all those disorders. So what exactly is Simons Searchlight doing to enable studies with iPS cells? As I mentioned in the very beginning, participants in Simons Searchlight can donate blood that is shipped and stored at our biobank at Rutgers University. The conversion of blood cells to iPS cells actually happens at the New York Stem Cell Foundation. And the resulting iPS cells are then shipped back and stored at the biobank at Rutgers. And it takes about 6 to 9 months to make iPS cells. All the iPS cells that are made with Simons Searchlight donated blood are available to researchers worldwide. The iPS cell samples can be requested through our database called SafariBase. And once a researcher requests the samples, we then review the request to make sure it's legitimate, and the samples are shipped to them. Safari has formed a collaboration with the Nancy Lurie Marks Family Foundation, short NLMFF, to finance the generation of iPS cells. And we think this will save researchers time and money and ensures that a high quality, technically homogeneous iPS cell resource is generated. And in the end, of course, we hope this will accelerate research to develop new treatments. Importantly, this would not be possible without your participation. Your or your child's blood donation makes this type of basic research possible. Thanks for listening, and I'm happy to answer any questions.
Hi, I'm Wendy Chung. I'm going to tell you this morning about some exciting advances we have in gene therapy. So your child or your family member's condition is genetic, and the obvious question becomes, can we do something to fix that? Can we go in and edit the gene? Can we fix the gene? Can we add back a gene? And many of you have been asking that question. I'm going to give you an update today in terms of where we are. I certainly am optimistic that in the future there are going to be many options in terms of genetically and molecularly based therapy, but really the million-dollar question is when. And I don't have a great answer to that, but I will say that there are many people working on this, and I'm going to try and demystify this a bit for you today by explaining some of the strategies. So on the left here is what we're showing as a cartoon of a normal gene. That gene encodes the instructions to make a protein. And again, on the left here is someone where that gene is working fine. Now, on the right, there's a gene that has a genetic change. It's shown as a broken gene here. And in some cases, it makes no protein. That gene just doesn't have instructions that work, so it makes no protein. In other cases, it may make a protein, but it looks different than the normal protein. And in some cases, this can lead to an individual with a genetic condition. And that's what we're going to be talking about today. So very broadly, depending on whether or not the problem with the gene is not making a protein or making an aberrant protein or a protein that looks different, there may be slightly different strategies. I'm going to go through the most common strategies that I think might work for your family member's gene, but to say that there are still evolving strategies. And so I won't have time to go over in all of the strategies or in great detail, but I'll go over the most common ones. So let's dig down for a second. So here on the left, shown in red, are individuals that have a gene that has this particular change in it, a change that may, in this particular case, result in disease, and it's because this protein is not made. In those cases, there can be different strategies, but strategies in which we add back a normal copy or a functional copy of the gene. This then makes protein that was missing or potentially not there in enough quantity, and then corrects the condition because we have added back what was missing. Now there's some subtleties in this. As we add back this functional copy of the gene, where does it go? Does it go directly within the genome or the genetic information of the person? Does it integrate or does it become part of them? Or is it something that's living off to the side in some way? And there are some important differences between that that have to do with whether or not we get it in the right place and whether or not it stays around for a long period of time. So those are some subtleties, but this is the basic idea. Now, in certain cases, I showed you in that picture, there may be a gene with a change depicted here. And in this case, it actually does make the protein, but in this case, the protein is not doing what we need it to do and may in fact disrupt or interfere or gum up the works of the regular gene. So it's doing something that we don't want it to be there doing. And in that case, we actually have to get rid of it. It's not simply replacing it or adding back, but it is actively getting rid of this red protein here. And so there are strategies. I'm going to use the term gene silencing, but it's one way or another of being able to knock down or get rid, and if we can, of doing it specifically for the form of the gene that's making the protein that we don't want to have there. And so in that case, the reason why we get correction is we are getting rid of this particular protein. Now, for doing that, there are different ways of being able to deliver what look like these little, I don't know, these little green ladders here, or half of a ladder. There's a way of being able to deliver it to the cells in various ways. I'm not going to focus on those details today, but it is critically important to know the difference between the case I described over here, where the gene, or the protein rather, is missing, and the case over here where the protein is present and we don't want that version of the protein there. Fundamentally very important to differentiate. And I'm glad to say that in the cases that we're going to be talking about for your gene, I think we're to the point of understanding those differences. So that's the good thing. Now, some of you have probably jumped to the obvious conclusion to say, well, why don't, if the gene with the change is here, just fix it? Like surgeons, just go in and snip it out, take out the version that's not working, sew in the version that does work, edit it, correct it, make it whole again. And if we fundamentally correct the DNA itself, then it will correct the protein. We don't have to do anything fancier than that. We can just go in and get to the root cause or get to the heart of the problem. And so this is a technology people are using called gene editing. You may have heard the terms CRISPR, Cas9. Those are effectively like the molecular scissors to be able to go in and to find just the right place, just the right location to do the snipping and then to do the sewing in. This is certainly a technology that is evolving very rapidly. Some of you have heard about the improvement recently called prime editing, which increases the fidelity or the likelihood that the editing gets done in just the right place. Now, the opposite of that would be some editing that's promiscuous, where you don't get to just the right place, but you may get to someplace down the road that you didn't mean to be editing, or someplace down the road over here that you didn't mean to be editing. And there is the possibility of misediting, actually putting in something or cutting and pasting in the wrong place, and that could actually be bad. That could actually disrupt a different gene that you weren't intending to touch and cause its own independent problems if it's not very precise, very accurate. So within this, as I said, there are more and more tools that are becoming available. Some of these use what we call vectors, or ways of being able to deliver these tools to cells. I'm going to be talking about that in just a second. Some of them are delivering these tools, these scissors, the thread to be able to sew this in. And really, the good news is that as these tools are developed for one condition, they can be readily applied oftentimes to other conditions. So you may be hearing about methods that have to do with gene therapy in the eye, or gene therapy for sickle cell disease, or gene therapy for a liver disease, or a bleeding condition. The good thing is that we are going to shamelessly beg, borrow, we won't steal, but we will borrow from others in terms of as they develop those tools. We will use those same tools that we're thinking about for your family's condition as well. So we do have to be patient. And what I want to give you is a little bit of historical perspective. You don't need to read all of the details here, but simply to show the timeline. So when we first started thinking really in the modern era about gene therapy was about the time I started my career in genetics, about 30 years ago in 1989. Now, the good news is we've come a long way in the last 30 years. We've made major advances in being able to understand this, but we also have learned some very valuable lessons. And I'll just point out one valuable lesson we learned in 1999 was that this is something that could actually do harm, that we need to be careful about this because a young man by the name of Jesse Gelsinger was actually unfortunately passed away during his gene therapy for a different condition. From that, we became, as a community, internationally cautious, excuse me, in terms of being able to be careful, be safe, have checks and balances, test things in cells, test things in other animals, but really to make sure that we were going to do no harm, that we were going to protect the safety of the individuals who went through gene therapy studies. And because of that, we've slowed down. We're not running quite as fast, but on the other hand, I can assure you that we're always have the participants' best interest in heart as we're taking those precautions. So let me tell you a little bit about how we get those genes into the cells. And there are some limitations based on these delivery mechanisms or these delivery systems that we have. So the first is that there is a virus called adeno-associated virus. And I'm showing you here this viral particle. Within this is something that is a virus that is part of what causes the common cold. We have done things to these, genetically engineered them so that you can't pass them from one person to another. So it's not like the cold, it's not like SARS-CoV-2. This is not something that gets infected from one person to another. We have basically paralyzed them so that can't happen. But they are good delivery systems that inside this little squiggly here is the gene that we're trying to insert. And so as we're doing this, as I said, we have inactivated these viruses. Importantly, this is a cell that I'm showing here, and this is the nucleus inside the shell that houses the genetic information. And these are meant to be chromosomes housing the genetic information. When this virus goes in and infects the cell, this genetic information for the new gene comes into the nucleus, but importantly, it does not integrate within the person's genetic information. So we call these non-integrating. By setting them up so that they are not integrating, they will not disrupt other genes that the person has. And one of the types of genes that we worry about in terms of disrupting are genes that are mission critical to the way your brain works or your heart works, and also genes that might protect you from cancer. We don't want this to result in changing one condition or trading one condition for another. We also have engineered these cells or used particular adeno-associated viruses that they hone in to particular parts of the body. And in fact, it's been quite successful, remarkably successful, at being able to hone into brain cells or to neurons. And that's been a huge advance. This is also something that works quite well for cells like brain cells, like neurons that are no longer dividing. Otherwise, if they were dividing, you can imagine this DNA that gets in would be diluted out over time. That is, that when this cell divided, you'd still only have one copy of that DNA, and therefore only half of the cells that would retain that DNA and make the protein. So because brain cells don't divide, this is a very good strategy. It stays there. As we have gone through these gene therapy trials, and this is true across many different conditions, part of the reason why it takes so long is because we are, as I said, trying to make sure that safety is paramount. It's a very, very important thing to us. And as we do this, we try and make sure that we get just the right dose of the virus. We don't want to give too much to the virus. We learned our lesson. We gave too much. Collectively, we as a community gave Jesse Gelsinger, the young man who died, he got too much of that virus. So we're very careful about that. We're careful about testing, as I said, in cells and what we call preclinical testing in animal models. We test to make sure that the gene gets there at high enough dosages of the genes, and then ultimately not just that it's safe, but that it actually works. It's efficacious. It has the desired beneficial effect to slow down the disease or even to be able to reverse the disease. So a lot of thought goes into this to doing it well. Now the success, the good news is, and I'm just going to cut down to this bottom two lines, there are already two medications approved, two gene therapy medications approved for two different conditions, one an eye disease or a retinal disease, another for a condition called spinal muscular atrophy or SMA. And I'm very happy to say I've personally been part of these SMA studies, and we can now treat babies, newborn babies in some cases, with the gene therapy. It's literally an intravenous infusion through a vein. We don't even have to do a direct injection into the brain. We can just give it into the vein as a one-time treatment. I hope that this is going to produce a durable effect, that this is going to be something that's going to last for decades and hopefully a lifetime. We're still only a few years into this, but it does already show to be quite promising. Although I'm not going to go into it today, there are many, many other conditions that are currently being studied in what we call clinical trials. In other words, we're still testing them. Other brain conditions, so importantly here, adrenoleukodystrophy, another brain condition that I think is going to give us significant experience in terms of how we can do this for neurological diseases. So coming to wrapping all of this up, there are many things that we consider, and we try and matchmake. We try and find for any one disease, understanding the mechanism of how does that protein have its effect based on the way that that protein is working or not working. Think about exactly what type of technology would be useful to be able to get the correct version of the gene in, be able to permanently keep it within the brain cells, and to make sure that it's safe. Now, as we're doing this, there are a few other additional things that we consider. We want to be able to get in just the right amount. So it's kind of like Goldilocks and the Three Bears. We don't want it to be too much. We don't want to be too little. We want to be just right. And that just right, we hope, is what's going to be both safe and efficacious. And that's still part of what we're working on as we're getting into brain cells, making sure that we're getting just the right amount, making sure that we're getting in the brain, we're uniformly getting it into all the cells across the brain. There may be some consequences if it's a patchwork, if it doesn't look like we get into all the brain cells uniformly, we're not sure what that impact is going to be. And so we do need to be thoughtful in terms of how to do this, as I said, evenly, make sure it lasts, make sure there are no adverse consequences or no complications. So as we're doing this, just to let you know, there have been folks that have been at this for decades. The good news is that I can feel a real acceleration in terms of the breakthroughs that we're having, being able to learn from other diseases, other neurological conditions, and importantly, the safety that we now see in terms of the types of vectors we're using, the viruses we're using to deliver this, and importantly, that this is something that looks like it is lasting so far. I'm getting, for all of us, excited that we need to get ready. We need to prepare so that when some of these breakthrough technologies are able to scale and be applied to other conditions, I want our communities to be front and center. I want us to be at the starting line, ready to go, ready to take off on this race. And so many of the things you're hearing about at this family meeting are how we as a community can prepare for that day. I don't know whether that day is going to be in 2021 or 2025. It's hard for me to say, but I do think it's going to be here much sooner than we might think, because we have made so much progress over the past 3 decades. So I'll stop here, but enjoy looking forward to the discussion.
My name is Paul Wong. I'm a member of the science team here at Safari, and by training, I'm a developmental behavioral pediatrician. I used to work with hundreds of families who had children with autism, with intellectual disability, and with genetic disorders. And I also spent a little bit of time in the pharmaceutical industry and at a biotech company. So today's topic, clinical trials, is something that is really near and dear to my heart. I'll be talking about what clinical trials are, why they're important, how they really work, and who participates in them. So to start with a 30,000-foot view, clinical trials are part of the larger drug development process. That whole process can be a very long and complicated one, and it starts with understanding the disease. This is work that probably a lot of people listening have actually contributed to by donating their blood, by showing up in research labs for studies of learning and memory and other aspects of the condition. So that's how drug discovery starts, by really understanding the disease. A lot of it happens in laboratories, and the next step is called target identification, where the researchers figure out exactly what kinds of cells, brain cells, where in the cell, what proteins or enzymes in the cell, what neurotransmitter receptors really need to be targeted. And once they figure out what needs to be targeted, then they go on to developing lead treatments, a lead chemical, a lead drug, or sometimes it may be a genetic therapy, and optimizing that lead. Even though it's not shown on this slide, a lot of the work here is also going to happen in animals and in cells or stem cells or something like that, to try to see if the lead really works. And then in the animal studies also to see what the side effects are, what kind of safety concerns there might be. And it's only after the potential for a drug working and safety are established that you move into clinical trials. And that's what I'm going to focus on for the rest of this talk. You'll sometimes hear about phase 1, 2, and 3 clinical trials. That's sort of the natural evolution. You start with fewer people in the study and then move on to more and more. You start off trying to figure out what dose is the best to work with, and then you try that dose in a larger number of individuals, etc. But we're going to talk generally about clinical trials, and all of that is necessary before you can get approval from the regulatory agencies like the FDA or the EMA in Europe. So what is a clinical trial? A clinical trial is a type of research. Specifically, it's a study that's actually conducted with patients, with people, to evaluate a medical treatment or a drug or a device. Here, of course, today we're talking about medical treatments, whether those are pills or a gene therapy, perhaps. The goals of a clinical trial include determining the best dosing, assessing the safety, in other words, what are the side effects, what are the adverse events that the drug might cause, assessing its efficacy, which means figuring out, does it really work? And sometimes a clinical trial will compare 2 different treatments to see which one works best and which one has fewer side effects. Why are clinical trials needed? In short, they're necessary to really prove that a drug works and that it's safe. And believe it or not, even though I won't go deeply into this, 100+ years ago, there was no requirement for a drug to be proven safe and effective. People literally sold snake oil, didn't work, wasn't safe, but you could sell it. And over time, the FDA was established, and then early on, FDA said, okay, you have to prove that a drug is safe. It wasn't until 1962, actually, that the regulations were put in place so that the FDA could demand proof that a drug was efficacious, that it worked. And today we're in a situation where you do get extensive testing in cells and animal models to look at safety and to look at efficacy, but you still need human testing because what happens in the laboratory doesn't always translate to what's going to happen with real life patients and what's going to happen with children. So that's why clinical trials are needed. How do clinical trials work? Well, at a high level, you could say it's pretty simple. You recruit subjects in, you perform assessments to see what their condition is like at the baseline, start the drug, and you perform follow-up assessments to see if anything got better and to see if there have been any side effects. Sorry for the misnumbering there on number 4. It should obviously come in between 2 and 3. You perform the baseline and then you start the treatment. And if you diagram it out, it might look like this. Patients get screened. You do that efficacy baseline to see what the symptoms are like before starting treatment. Then you start the drug, some dose adjustments, do some phone calls or video calls to see how things are going. You might have the subject come back in for an evaluation a few weeks later, follow up with more phone calls again, see how things are going, then bring them in at the end of the study to see, okay, now after 3 months, 6 months, whatever it is, do we have improvement? Is there efficacy? Does the drug work? After that, some drugs can be stopped immediately. Some drugs have to be slowly tapered down. You have phone calls again and a follow-up appointment just to make sure there are no side effects that you missed earlier on. So that's sort of how it looks in general. But if you drill down, it can get really complicated. So each of those steps really has a lot of smaller steps. Recruiting patients isn't just like snap a finger and they show up. You have to advertise, you talk to people on the phone, tell them what the study is like, review the medical records. They have to travel to the sites where the study's happening. Then once they're there, you still have to figure out if they're really going to be eligible or not. You do a physical exam, take a medical history from the child, the parents. You look at other records that they have, physical exam, doing blood tests, make sure everything's okay there, et cetera, et cetera. So all in all, there can be really a lot of little steps in there. This table is taken from a study that actually was done, and it sort of just lays out all the individual things that happen at visit number 1, visit number 2, all the subsequent visits, and they happen monthly initially, spacing out to every 3 months. Got things going on like an x-ray, a history, physical exam. You have blood work as well to make sure blood count is okay, your blood chemistry is okay, and then later on you got to repeat that at all those visits to make sure there are no side effects from the drug that only show up on blood tests. And all sorts of different assessments. In this case, there were examinations of muscle strength. There was examinations of the heart to look at side effects there. This was actually a gene therapy study, so the gene transfer actually happened at visit 2. But what I want to talk about a little bit more today are the actual assessments, because that is really a central part to every clinical trial. So when we figure out, when a clinical trial is being planned, obviously a key thing is figure out what is going to be assessed. Are you going to be looking at memory in the subject and how memory improves? Maybe you'll look at speech. Maybe you will look at other kinds of motor skills like walking in a young child who has a motor delay. Maybe you're looking at social function and how an individual interacts with their peers or with their parents or other people. You could also be looking at repetitive behaviors. You could be looking at various aspects of health. But of course, these questions are pretty complicated. One of the factors that complicates what you're looking at is age, because motor skills are going to differ for a 9-year-old or a 10-year-old as compared to an infant. Social skills are going to differ for a teenager or a young adult compared to a preschooler. So you have to take all of that into account as you're figuring out how you're going to do the assessments. And of course, there's individuality. Everybody's different. And even if 2 people have the same genetic diagnosis, that doesn't mean they're exactly alike. There's going to be variability. One person might show more speech symptoms, one person might show more difficulties with memory, and a third person might have difficulties with fine motor skills. So assessment is a really important part of every clinical trial, figuring out what you want to look at and figuring out exactly how to do those assessments. Here's an example from a clinical trial of a gene therapy that actually happened a couple years ago. And this was a trial in a condition called spinal muscular atrophy, SMA, which causes really severe challenges to motor behavior, to motor function. Infants are affected and they can become extremely, extremely weak. So on the one hand, you might think, oh, we'll just ask, is the baby sitting yet? Is the baby walking yet, or just standing? But actually, you have to look at a much finer level, much finer resolution than that. So for this study, the scientists created a brand new scale. They called it the CHOP in 10. You can see what it's an abbreviation for. We'll just look at item number 10 as an example, knee extension. So lifting up the bottom part of your leg. And they tell you exactly how you do this. You put the infant in your lap or sitting over one edge of a mat with the head and trunk supported, and your thigh is horizontal, like you're sitting on a chair. Then you tickle the foot of the infant or gently pinch the toe. What does the baby do? Can they extend their knee? Can they lift up their lower leg to 45 degrees? If they can, they get 4 points on this scale. Can they extend it not quite to 45, but 15 to 45 degrees? That would be 2 points. Any knee extension, but less than 15 degrees would be 1 point. But sometimes actually this condition causes such profound weakness that the baby cannot extend their knee at all and they get 0 points. So this scale had something like 20 items. Knee extension is only one of them. But you can tell it's like this really finely graded assessment of motor abilities. And here's the results that they saw in this trial, which were fantastic. Again, this was a gene therapy trial for this devastating condition. The treatment was administered very early in life. And each of these lines shows what the motor abilities were like for a single child. So for example, this line, I think that's maybe patient number 6. They started out at about 1 month old. They got a score of something like 14 or 15. And then their abilities got better and better, so that by the time they were 20 months, so a little bit more than a year and a half, they got a score of 50, having started at 15. And you can see there's variability. Some kids started out better than others, some kids started out lower than others, but almost every child who got this treatment showed improvement in their motor abilities. Great result. But if improvement happens, can we be sure that it's really due to the treatment, due to the drug, due to the gene therapy, or whatever? Unfortunately, the answer is no. We can't always count on it being due to the treatment, because sometimes people seem to improve no matter what happens, or we think they're improving no matter what happens. Here's an example from about 20 years ago or so. There was a lot of excitement that autism might be treated with a drug called secretin. Secretin was given IV, and here what we saw is that people who got secretin, their symptoms decreased. So that's good for autism when your symptoms decrease. From the baseline to day 1, day 2, and all the way out to week 4, symptoms were much less than when they started. But in this experiment, they also treated some patients just with placebo, something that we know doesn't work. Could be a sugar pill, let's say. You know it's not doing anything for autism. And even then, there was improvement in those patients. They didn't know whether they were getting secretin or placebo. It was a so-called double-blind study. The families didn't know, the doctors didn't know either. Why did that happen? We don't really know why these placebo effects happen. Sometimes people only enter a study when the symptoms are really bad. We know that symptoms can change from day to day, week to week. So maybe they came into the study when things were worst, and then, of course, things naturally vary. So when they looked a couple of weeks later, things were better. There's another example I just want to share with you here. Here's another study where actually no treatment was given. Everybody knew no treatment was given. Nobody got a pill, nobody got an infusion or anything like that. They just asked the parents, okay, come in, rate your child's symptoms. This is autism again. And then they had them come in 8 weeks later at time 2. Everybody's symptoms improved. Everybody's symptoms diminished. They got better, even though they didn't get a treatment. So what's happening here? We don't totally understand what causes these kinds of changes. But the point is, just because someone gets better, we can't be sure it's due to the drug. At least in a large majority of cases, we can't be sure. And that's why placebo is needed in most clinical trials that are done. once in a while, when the symptoms are so severe and we can be confident that nobody's going to get better without an effective treatment, once in a while when that happens, then we don't need a placebo and we can go with what's called a natural history study. So I showed you this picture before. This was the gene therapy actually for spinal muscular atrophy, where almost everybody who got the treatment got better, even though there was some variability. Was this due to the drug? Yes, it was, because we know that the natural history of spinal muscular atrophy is that everybody gets worse. Natural history just means what happens if you don't do anything, if you just let the condition go naturally without any treatment. What do you see? This graph looks different. The lines are thinner, slightly different colors. There are no points at the different time points, but I actually have scaled them the same. And this is what happens when SMA is not treated. Everybody gets worse. Again, there's variability, but you can see nobody in this right-hand picture looks like hardly anybody in the left-hand picture. So when the natural history is so dramatic, then we can be confident improvement is due to the treatment, and then we don't need placebo. So last slide here. Who participates in the clinical trials? Well, many of you who are listening to this, many people who are Simons Searchlight families are going to be asked to think about participating. And the key points I want to leave you with is, first of all, it's voluntary. Nobody has to participate. And in fact, even if you start in a trial, you can withdraw at any time. Your decision to participate or to withdraw will not affect the rest of your medical care, will not affect your participation in Searchlight. If you are considering participating, be sure to just get all the information that you can from the study team. They're going to give you printed information. They'll answer your questions, ask every question that comes to your mind, and talk to whoever else you trust, your personal doctor, your geneticist, other trusted advisors. This is a very important decision for you to make. Only do it if you're comfortable with it. But know that if you do participate, your participation will be very highly valued. So that's all I have for today. I'm sure you're going to have a lot of questions. I hope that there will be an opportunity to think about whether you want to participate in a clinical trial. I just want to leave you with these 3 other sources for information that might be of help to you from the American Academy of Pediatrics, from the FDA, and from the NIH, the National Medical Research Labs down in the Washington, D.C. area. Thank you very much.
All right, so now it is time for the Q&A with all of our speakers. So just as a reminder to everybody on the call, if you'd like to submit a question to our presenters, the Q&A box is at the bottom of your screen. It looks like little talking bubbles. And when you submit questions, if you can try to direct them to a particular presenter or refer to a presentation topic, that would be very helpful. And with that, we'll get started with the first question, which I believe is for Dr. Chung. My son has Hive P2. He's one of only 2 children with mosaic Hive P2. Would it be important to have research done on his genes compared to a child who is fully affected? How do I get a researcher excited about our children to take on this research?
Great question. So let me frame this for some people who may not understand the question even. So the point about mosaicism is that for this particular child, all the cells in the body don't all have the same Hive P2 change. So just like you have tiles that are mosaic, in the same way the cells are mosaic. Your son is incredible. So as a resource, let me try and explain and put some pieces together with what Dr. Summer was talking about. We can take cells from your son and be able to have sort of the perfect, if you will, resource for the community, because some of the cells we can fish out to be the ones that have the Hive P2 mutation, and we can fish out and identify some of the cells that do not have the Hive P2 mutation. And so this is the absolute perfect comparison to make as we're trying to understand what the Hive P2 change causes within those cells to be different, and what medications or what treatments might be helpful. So we're glad if you want to reach out to us back channel. I'm not sure, because we're not exchanging names here, in terms of whether or not your son is already in the collection that Dr. Sommer talked about, but I'm sure researchers and the community in general would be incredibly grateful for the contribution that your family very uniquely is able to make. Within this, I will say the other thing that gets confusing is that individuals that are mosaic, because we obviously don't take biopsies and look at their brain or look at other parts of the body, we're not sure exactly how many of the cells, particularly in the brain and different parts of the brain, are affected. So it's sometimes hard for us to know that association between severity of different symptoms and how that matches up to what's going on in the body. So there are some particular challenges for individuals who are mosaic, but we are interested in trying to understand that as best we can.
Thanks. So the next question is to Dr. Green-Snyder. You said reports are done based on our survey answers. Have there been reports in the past, or do we need to wait for the website to change over, and will they be available soon?
Yes, so can you hear me?
Yep.
Yes, we can.
Okay, thanks. So I don't have a specific date for you, and we can check on that. Reports were available in the past. Our first step was to try to reload the reports that were available in your old accounts before we changed providers for the website. There were reports there for the Vineland Adaptive Behavior Scales, the Social Communication Questionnaire, and the Social Responsiveness Scale, if you completed them. So the first step was to bring those over, and the second step is to generate new reports for any brand new surveys that you may have completed more recently. So that's where we are right now. Again, we'll have to reach out to you all to let you know the date that that's going to be ready, but we will be able to give you access to those.
Great, thank you. And so the next question is for Dr. Chung. Are there any developments in the works for treatments that could possibly target the MED13L mutation? And if so, or sorry, that's the question.
So for MED13L, I don't know of anything specifically. There are things that you're going to be hearing from the meeting this weekend. And so there may be even things that I don't know that are going to come up this weekend. But no, I don't know of anything specifically in terms of a clinical trial that's coming up within the next 6 months or a year.
Thanks. Sorry, just reading through this question. This person is saying they're interested in being tested and eventually having gene therapy for their son who has HIFP2, and the mother is mosaic. And they just want to know, how could they apply to therapy or apply to be studied?
So perhaps I'll take this for Dr. Wong and myself, which is that we're very committed to make sure that as soon as there are clinical trials, whether they're gene therapy or anything else for any of the groups, that this information is conveyed to the family group leaders. In the United States, there's a website called clinicaltrials.gov. Again, and we can distribute this to the group afterwards, but clinicaltrials.gov, G-O-V. That always talks about, or is a way to be able to see and get updates on any clinical trials that are available for any condition. It doesn't have to be just for Simons Searchlight, but specifically, the family group leaders are very good at being able to disseminate information specifically about upcoming studies, treatments. We have a program called ResearchMatch that we'll use to disseminate information to anyone who's registered in Simons Searchlight. So again, if you are registered in Simons Searchlight, we'll have that as another communication channel to get information out to you as well.
Great, and I think this might be another question for you, Dr. Chung. This person said that they're trying to sign up for Simons Searchlight, but they're stuck at the lab report step. All they have is a letter from their geneticist. And they want to know, what exactly does the lab report tell you, and will you share this information with your participants and findings about this individual?
Sure. So we had a talk actually last Friday that went over how to read a lab report. I am now more cognizant of people getting stuck at the same stage. So if that was the case, do me a favor. We'll be sending out some messages, but also we will take that letter that you have, even though it's not an official lab report, that may be a letter, or what I often call a results letter from your doctor. We can do some work on our end to fill in the gaps about what's missing from the lab report. There's some very technical things that come in the lab report that allow us to verify that the variant is actually what we think is the cause of your family member's symptoms. And so it makes it easier if we have that fully there, but realizing now that not everyone has that information and wanting to be able to give everyone the opportunity to participate, we'll try filling that gap. And if necessary, we'll also have you sign a medical release form so that we can get that directly from your doctor. But we understand and we'll help you get through this whole process.
Thank you. Another question for Dr. Chung. What companies and what specific trials look the most promising for gene therapy and gene editing?
So that's a good question, and it's complicated. And the reason I say it's complicated is in my talk, I started explaining a little bit that there's what we think of as mechanism for the gene and exactly how the gene works. If you're missing the gene and it just didn't show up to do its job, that's a different mechanism that if the gene product, when it's not functioning correctly, is doing something it shouldn't be doing. And if we need to get rid of that or kind of be able to silence it or suppress it. So there are several different companies that are working on different strategies. At this point, the good thing is there are over 20 companies doing some form of either gene replacement or gene editing or message editing, or being able to use oligonucleotides to change splicing or to suppress expression. A lot of different things going on. I just want to say I'm not advertising. I don't have any financial relationships or anything with that with any of the companies. But I do think there's, the way I describe it is there are lots of hammers and there are nails, and there are screwdrivers and there are screws, and there are wrenches and there are bolts. And part of what we need to do now as a community is figure out the matching of who's a nail and which hammer do we need to be able to connect in terms of those. And so we're still a little bit at the early stages in terms of understanding mechanisms for all the genes that we're talking about amongst the groups today, trying to understand what are going to be both safe as well as effective. And so I won't say that there's any particular front leader in terms of being able to do this. There are some that have been, I'll just, again, I'm not trying to promote any one company. Spark is a company, for instance, that was first out in terms of having FDA approval for a gene therapy for the eye. I'm not saying Spark is necessarily going to be doing other things, and I don't want to confuse people because people know that we also have a study called Spark. This is different. This is a company called Spark, but that is a company that has some experience in terms of being able to do this. And as I said, there are others, but we can certainly back, sort of offline, have some more detailed discussions for each of the groups.
Great. And for Dr. Chung, since many of these mutations in genes cause developmental issues at an embryonic level, is it even possible for treatments to be effective for functions that never did develop in the first place?
That's a great question. And Dr. Spiro was actually addressing some of that when he talked about the reversibility experiments in the mouse with Rett syndrome. So we don't know the answer to that. And in fact, one of the critical issues, we as scientists actually think about this a lot, and we talk about doing reversibility experiments, trying to understand which of these genetic conditions is like Rett syndrome and may have a longer period of reversibility than we might have even appreciated, and which ones really do have a smaller window, in terms of development, and maybe a critical period of development where we may not be able to have an impact long term. I guess the one thing I would say is that we don't know until we know. And so I think it's really, it's important for us to do the experiment. We can have guesses, we can have hypotheses, that's what we do as scientists, but we need data to be able to drive our decisions. And so we just need to do some of these experiments, actually a lot of these experiments, and come up with the answers. But it's a great question, and it's the way we strategically think about what to go, where to really double down and push harder on.
Thank you. And this question maybe for Dr. Green-Snyder, maybe for Dr. Chung, regarding blood donations. If a family member has an unrelated genetic condition like MS, does this affect their suitability to donate blood?
I'll let Wendy take that.
Okay, so for most conditions, the answer is no. The condition where it makes a big difference is if you've ever had a bone marrow transplant. So there are very few people who would be in that circumstance, but if you had leukemia, for instance, or if you had another condition where you received someone else's bone marrow, then getting a blood sample from you does not help us because it's really going to give us a blood sample from whoever gave you the bone marrow. Otherwise, in general, for most of the things that we're talking about, it's not a problem.
Thank you. And another question for you. If you study reversibility for one mutation, how do you know the results will be the same for all mutations, or do you need to do a reversibility experiment for each mutation?
Boy, you guys are like superstars in terms of your questions. So we don't always know that all mutations act by the same mechanism, but in many cases, we do know that we can bundle them together. And so we do know that certain mutations we think of as acting by a loss of function mechanism. That is that they don't show up to do their job. And we tend to cluster all of those together. If we made a mouse model that represented all of those that had loss of function, then we make the assumption that that extrapolates for that class. The thing that's a little bit trickier is with what we call missense mutations or single amino acid substitutions. Whether or not every single one of those acts in the same way, we're not always 100% sure. And then your point is a good one, that in some cases we might have to do more than one mouse model to test that hypothesis.
Thank you. And that is the last question we've received. So I just want to hold for a couple more seconds to see if there are any last minute questions that folks want to submit. Okay, so I'm not sure who to direct this question to, but how promising do you feel ASOs are for this community?
I'll take that and then other people can chime in. So these ASOs are allele-specific oligonucleotides. As some of us were alluding to, these are things that are made artificially, and we have to be able to get them into the brain. And so for spinal muscular atrophy, we use this by injections into the spine. For others, people have talked about injecting them into a reservoir in the brain. And the bottom line is they work very well for spinal muscular atrophy and in general for splice mutations, a very specific type of mutation. It looks like they can be effective. And I can say that the company that's working on this, Ionis, is looking at several different conditions, including conditions like Huntington disease, but also several other neurological conditions. I do think it has promise. There are certain advantages to using the ASOs. One is that the toxicity we understand pretty well at this point, and in general, the generation of ASOs are very safe. Whether or not they're effective, I don't think we always know, but at least they're safe. And they also have the advantage of being able to stop them. So in general, they last for about 3 to 4 months, but they don't last forever. And so as being able to test something and being able to pull back if we're concerned that there's a problem, that does give us that option.
Thank you. And the next question I think may be for Dr. Sommer. When studying mutations, can you get the same results from blood, skin, saliva, and urine?
Yeah, I'll try to answer that. So, and this may have been triggered by something I said in my talk, which was that these iPS cells that you can use then to make neurons from, you can make from blood as well as from skin cells. So that's one thing. You can use blood or skin cells as the source there. But it's true, you can also study blood cells or skin cells and culture them and look at how your gene affects the function of the protein that it codes in those cells. And there are some things that you can study in blood cells or in skin cells that you could equally study in both cell types. Now, urine and saliva, I think about a little bit different, because they're fluids that contain trace amounts, some cells in them. And I'm sure you've heard saliva with regards to genetic sequencing, because there are some cells in the saliva, like some cells from your cheeks. You can actually do gene sequencing and search for mutations in DNA that comes from saliva. But otherwise, I think you could look for metabolites in saliva, but it's not a particularly good source for cells to study the cells in there. Urine is similar. It's also a solution that contains, few cells, epithelial cells, and you can culture those cells, and people have actually made IPS cells from epithelial cells collected from urine. But again, it's much less efficient to do that because the amount of cells in there is much smaller. So, the answer is complicated. You can do some of the same things in all those sources by culturing the cells that come with it. But you can also do much more, for example, with the IPS cells, because you can create all these many different cell types from it and then study how a mutation affects the function of those different cell types.